Hi, Assalamualaikum. My name is Auni Shazana binti Zaha and my metric numbers is 23DBT19F1035. Today, I will be running the first lab, which is purifying protein by column chromatography. In this simulation, it will purify protein expressed in E. coli cell from a recombinant plasmid. In order to obtain the protein, it will first use lysis buffer to open the transformed E. coli cell. Before lysing the cell, it is important to increase the quantity of protein that interests, in this case, red fluorescent protein, RFP. The RFP is expressed from a recombinant plasmid that contains the RFP gene under the control of an arabinose-dependent promoter, as well as a gene for ampicillin resistance. A single transformed bacterial colony containing the introduced red fluorescent protein RFP gene is transferred to a flask containing luria broth, LB, and ampicillin and incubated at 37 degrees Celsius. This lets the transformed cell replicate, creating a large population of identical cells and increasing the amount of RFP protein expressed. The next procedure is pellet additional cell. Set the P1000 to 1000 microliters. Attach a tip and open the EC2 tube. To increase the amount of red fluorescent protein, we add more cell from the second tube. Draw up 1000 microliters of liquid culture and dispense the culture into the EC1 tube. Open the biohazard waste container and eject the tip into the biohazard waste container. Spin EC1 tube for 10 minutes and remove the EC tube. Open the EC1 tube and open the waste collection jar to tilt your tube over the waste collection jar. The third procedure is resuspend the cell pellet in EB. Set the P200 to 100 microliter and attach a tip. Open the EB tube and draw up 150 microliter of elution buffer. It is to release the hydrophobic proteins. Next, dispense the culture into the EC1 tube and eject the tip into the biohazard waste container. The fourth procedure is lyse the cell. Vortex the EC1 tube for 10 seconds to resuspend the cell pellet. It is to mix small volumes of liquid by agitating the tube. Attach a tip to the P200 and open the LYB tube. Draw up 100 microliter of lysis buffer and dispense the lysis buffer into the EC1 tube. After that, open the biohazard waste container to eject the tip into the biohazard waste container. Centrifuge the tube and set a timer for 12 hours to incubate EC1 tube overnight at room temperature. The next procedure is equilibrate the column. Place the waste collection jar under the valve of the chromatography column and open the column to drain away the liquid. Spin down the EC1 tube for 10 minutes and then remove the tube from the microcentrifuge. Set the P200 micropipette to 200 microliters and attach a tip. Open the EC1 tube and draw up 200 microliters of supernatant. Dispense the supernatant into the super tube. Open the biohazard waste container and eject the tip into the biohazard waste container. Next procedure is equilibrate the soluble fraction in BB. Attach a new tip and open the BB tube to draw out 200 microliters of the binding buffer BB. Dispense the BB into the super tube and open the biohazard waste container to eject the tip into the biohazard waste container. Apply the soluble fraction to the column. Pick up the P1000 micropipette, open the tip box and attach a P1000 tip. Draw out 400 microliters of the super solution to wash out proteins that are hydrophilic. Dispense the red super solution into the chromatography column. Open the biohazard waste container and eject the tip into the biohazard waste container. Place the waste collection jar back under the valve. Open the column and allow the liquid to drain into the waste container. Close the valve when the liquid is just above the resin bed. Last but not least, wash the column. Set the P1000 micropipette to 1000 microliter and attach a new tip. Dispense 1000 microliter of WB solution into the chromatography column to separate the different chemicals. Open the biohazard waste container and eject the tip into the biohazard waste container. Open the column and allow the liquid to drain into the waste container and close the valve when the liquid is just above the resin bed. Lastly, elute and collect the RFP protein. Using a new tip, draw up 1000 microliter of the EB solution and dispense gently down the side of the column. Repeat this step. Open the column by turning the valve. Watch the red protein traveling through the column. When it nears the bottom, close the valve. Place the open RFP tube under the valve. Open the column to capture the RFP. Close the valve and RFP tube and place the waste collection jar back under the valve. Dispense 1000 microliter of the column equilibration buffer CEB solution into the chromatography column. Open the column and allow the liquid to drain into the waste container. Close the valve when the liquid is just above the resin bed. Repeat 3 times before storing the column away.